Hi, I'm Larry Johnson. I am a professor here at Texas A&M University, and today we're going to continue with the introduction to microscopy. We'll talk about the pancreatic cells, we'll talk about types of staining, we'll talk about stomach cells and stains, different types of electron microscopy, observation of organelles at different magnifications, and different types of microscopy. Look at this summary and questions at the end.
So if we leave blood and go to the pancreas, you see the pancreatic acinar cells that produce pancreatic juices are located in through here, and you see the kind of blue at the bottom and then kind of red in the middle. We'll talk about that later. And then the clear cells in the center are the cells from the islets of Langerhans, beta cells, alpha cells, beta cells that produce insulin. So if we take a look at that, we can see the lighter staining is the intracrine part, and the acinar cells is the extracrine part. Namely, these secretions go in a duct, and these do not. If you look at hematoxylin, hematoxylin and eosin are the two stains that we use in combination, and the acid dye is eosin. So the acid dye eosin, it stains proteins red. So what we see here in the lumen in through there are proteins that are produced. They're the enzymes. And the basic dye is from hematoxylin, and it stains ribosomes and nucleic acid nucleus blue. So here you see the blue, the nucleus here, and the bluish tint to the cytoplasm is due to the high density of ribosomes in the rough endoplasmic reticulum. So therefore, the staining characteristics tell you about what's inside the cell. Is it more acid, which would be in the ribosomes and in the nucleus? Then it will be blue. If it's more proteinous, it will be red, like these granules. And we can see it very nicely here. Secretory granules are red as they are protein-rich in enzymes. That's what we see in here. So it's stained with the eosin. In contrast, the basal portion of the cell is blue. You can see blue here because it has a high density of ribosomes. So that's with H&E, but if you look with it with toluidine blue, everything's blue. And you have to look at the cells at varying intensities. The intensity tells you about it. You can see the dark granules in through there that we saw in the previous one. The granules are located in the lumen, clear cytoplasm in through there. You go by the shape, size, and the darkness indicate the density of the tissue. So everything's blue here, as opposed to hematoxylin is you look at tinctorial or color characteristics to tell you what's there. But here you have to look at shape, size, and darkness. So if we look at hematoxylin and eosin stain versus toluidine blue versus electron micrograph, we can see these granules are indeed these dark granules that you see. Here's a lumen in through there, so the cells getting nutrients from the bloodstream down below, and cells are processing things and emptying them into this lumen, and that's part of the extracrine component of the pancreas. So these are secretory granules that you see there. You can also see them in the toluidine blue. So you can see the granules are located right in through here, right where they are here in the hematoxylin and the eosin. And you see, so those are protein, eosin staining in the case of H&E. Also down in here you see rich swirls of things. This is a rough endoplasmic reticulum. So it's the ribosomes on them that gives us blue tint to it. So blue tint, like the nucleus, is blue because of the nucleic acids that are in the ribosomes or in the nucleus. So what we see there with the toluidine blue, all we see is kind of smooth cytoplasm, and that is the rough endoplasmic reticulum with the ribosomes. Another tissue we want to look at is the mucosa of the stomach. So the mucosa is the epithelium lining the stomach. It is the lamina propria and the muscularis mucosa. So those three things make a mucosa. And we'll look at the mucosa. Here's the mucosa. And this is a submucosa, connective tissue of the submucosa. So if we look at the mucosa, we can see a couple types of cells. Chief cells produce pepsinogen, and parietal cells that produce hydrochloric acid. So you can see these big cells here are the parietal cells, and the one is more bluish tint with these vesicles in through here are the chief cells. And so they produce two different things. And here we can see them again. These are chief cells and the parietal cells. Now, if we look at those same cells with toluidine blue, we can see the secretions of the chief cells, but also on the parietal cells, we can see there's granularity in there. This granularity is the mitochondria. So you can see mitochondria in there. So here you can see the chief cells, the secretions in both of these, very nice. And then in the parietal cell, we can see these dots in here. See these little dots around here? All these are mitochondria. Mitochondria is important in transport of hydrochloric acid outside the cell. But chloride one way and hydrogen is also being pumped out. And so here we see some nuclei. This is the nucleus of a chief cell, nucleus of the parietal cell, and a nucleus of a fibroblast that surrounds there. So the toluidine blue will let you see some cytoplasmic detail. In this case, we can see the mitochondria that we cannot see with H&E. The other tissue is the testis, and here we see the capsule, we see the seminiferous tubules, and in between there are Leydig cells that produce testosterone. And here we see the Leydig cells in through there. We see some nice fibroblasts in through here. We can see a lymphatic endothelial cells. We can see a little, this is probably an arteriole, little arteriole, maybe a bigger one, and then a host of germ cells. Some are round and some look like bullets. Now, just to illustrate the fact that tissue is transparent, and you have to stain it. The reason you stain it is to stain it differentially, so you can see some different components in there. But as a rule, tissue, if it's not stained, is transparent. For example, we're looking through the cornea of our eye, and if it wasn't transparent, we wouldn't be able to see through it. But mostly tissue, unless it's specifically pigmented, is transparent, and you have to stain it to be able to see it. Also, this staining facilitates you to observe cells, but these cells are all dead. So they're dead already. And we're using bright field microscopy to observe stained cells. If you want to look at live cells, you need to use phase contrast microscopy or differential interference contrast microscopy, a different type that we're not using. So here you see an H&E of the testis. You can see the bullets of the more mature spermatids. You see these primary spermatocytes. But take a look at these Leydig cells in through there. There's a nucleus of one we see with toluidine blue, and you can see lipid detail, lipid inside here, which you can see little white spots here, but you can't see the cytoplasmic detail with H&E that you can with toluidine blue. Toluidine blue is what people use before they to locate what tissue they're looking at with electron microscope. And electron microscope has different images that you can see, and what you want to be is a little bit mindful of magnification that you have there. When you think about magnification of a light microscopic level, think about a red blood cell. You can always see a red blood cell in a tissue somewhere, and it should be about 7 micrometers. Use that as a gauge to determine the magnification or relative magnification. In the case of electron microscopy, a good thing to use is ribosomes. Ribosomes is a good thing to indicate that you can use mitochondria as well. So what we have here is a lowest magnification going to the highest magnification. Here we can see the lipid bilayer in the membrane. We cannot see it over here. So if we start with a 90,000 magnification, we can see these neutrophilic granules that we talked about before, the monocyte. We can see mitochondria that are in. We can see some granularity in these lymphocytes, but you can see mitochondria for sure. If you look at these cells, these are polychromatic normoblasts. These are precursor to red blood cells. You can see the nucleus there. And you can see ribosomes in through there. We see mitochondria a little larger at 13,000. We go to 60,000. We can see mitochondria quite a bit larger. And you can see ribosomes very distinctly here. This is in the retinal plasma reticulum. There's the Golgi up there, but the mitochondria is more defined. Same magnification, we can see microtubules, microfilaments. But we can see the mitochondria, and here's the ribosomes of the polyribosomes in through there. If you jump to 80,000, ribosomes are quite bigger. And so the mitochondria are larger as well. And finally, if we jump to 200,000, you can see the lipid bilayer. You can see the two layers, two dark and one light in the very center. You can see microtubules. This is a centriole with the microtubules. So if you just look at mitochondria, you can see they increase with magnification. So a different view, different details. And then if you look at ribosomes, you see their increases. The ribosomes here much bigger than those and those based on the magnification. Also, there are different types of microscopy. Usually you talk about transmission electron microscopy, you're talking about conventional transmission electron microscopy that we've seen so far. This is not. This is carbon replica electron microscopy. It's good for surface detail. And here we see the zona occludens, where two cells are attached together. This is another carbon replica, and this is a lymphocyte. You can see the nucleus, you can see nuclear pores, you can see the mature face of the plasma membrane. This is a different type too. This is scanning electron microscopy, good for surface detail, as you can see. This is scanning EM of a kidney. This is a nephron, a glomerulus that we see. And so in terms of conventional transmission electron microscopy, this one and that one that we saw earlier is conventional. These two are scanning electron microscopy, and these two are carbon replica. And so in summary, remember, use your atlas to be able to identify the different structures that we see because they're labeled already there for you. Use your atlas is very important. One question that we want to ask, which type of microscopy or staining is better for observing cellular detail? Light microscopy with toluidine blue, with H&E, or transmission electron microscopy, typical EM staining? And the answer is toluidine blue can see more detail, certain more detail in cytoplasm than you can H&E, but transmission is by far has a higher resolution, be able to see more detail. So here is the Santa Ana Canyon in Big Bend, Texas National Park in Texas. Over here is Mexico, and over here is the United States.
We want to thank the various papers and publications, textbooks, from which we modified drawings, pictures, and images from. So that hereby acknowledging that these are the original source of those micrographs that were images, drawings that we used in the presentation. So here we acknowledge the original source. Thank you. So that's the end of our introduction to microscopy, pancreatic cells, types of stains and cells in the stomach, different types of microscopy, different magnifications as organelles look at different magnifications, summary, and the one question. Thank you.